I'm going to introduce you in French, but vous comprenez bien que Jeremy, bah, vous comprenez peut-être pas, mais il est anglophone de McMaster à Hamilton. Il va apprendre le français s'il est dans le milieu à l'avenir, mais pour l'instant, il va présenter en anglais. Jeremy est un prof adjoint sous promotion à la prochaine étape. On verra si ça marche cet été. Je pense que oui, entre nous. Il a fait son postdoc avant de commencer son premier poste à McMaster, son postdoc à l'Université de Colombie-Britannique à Kelowna avec John Little. Et avant ça, il était à l'hôpital pour enfants à Ottawa. Il a fait son PhD et sa maîtrise à Queen's à Kingston. Son intérêt aujourd'hui, c'est de vous parler de l'exercice, les cétones et la préservation ou la construction. Il parle to improve brain health, to construct a healthier brain. L'idée, c'est d'essayer d'améliorer la résilience du cerveau lors du vieillissement. Son intérêt pour les cétones nous touche pas mal dans mon sens, dans mon équipe, parce que j'ai la chaire de recherche en cétothérapeutique, donc les cétones, et un des objectifs de son embauche, si ça marche, c'est qu'il prend la relève pour continuer ce projet-là. Donc, Jeremy, over to you. It's a pleasure to have you back for your second visit with us. This time, he's meeting vous autres. He's meeting les gens en division de neurologie en médecine et sa conjointe l'accompagne pour voir si c'est vraiment ici que peut-être ils vont prendre la prochaine étape de leur vie ensemble. Alors, Jeremy, over to you, and perfect. We're looking forward to hearing you talk.
Bonjour. Thank you, Stephen, for the intro. As Stephen mentioned, my French is not that great, especially for speaking. So I appreciate everybody's attention in English today. It will be a top priority as well to become not just proficient, but also fluent and engaged in French language as well. So thank you very much. As Stephen mentioned, we're here considering the chair of keto therapeutics and the potential of me joining the team here. My wife and I are both visiting to check out Sherbrooke. So we got out to Orford yesterday, did some snowboarding, quite enjoyed it, and then went to Magog as well and enjoyed some of the breweries. So it's a really lovely place you have here. I want to give you a background about myself very briefly and what my lab team looks like at McMaster University. I was hired in 2020 in July, which is a fantastic time to become a human physiologist running human studies. So I actually lived an extra year in Kelowna and taught remotely. So really my lab got started in September 2021. But now we're a big group of PhDs, postdocs, and master's and undergraduate students. The real goal of this research, though, is to look at some of these mechanisms by which exercise, exogenous ketones, and behaviors like sleep or cognitive training impact brain function. And really, for me, the goal is to understand how mechanisms work to then build interventions that we can use to optimize brain health. So really understanding how to trigger brain health benefits. In our lab, what we measure is brain blood flow and cerebrovascular function through a variety of techniques. We've been using MRI, especially in our trials recently. We measure cognitive function as well as different biomarkers. And I'm particularly interested in a neurotrophin called brain-derived neurotrophic factor, or BDNF, which is thought to be this miracle grow for the brain, although it's not the only important factor. And then finally, interested in cardiometabolic health and function. So my training is as an integrated exercise physiologist, but starting to bring a lot more RCT design and implementation in clinical populations as well. Just to give you a view of what it looks like in our lab currently, we use ultrasound techniques, both linear ultrasound as well as transcranial ultrasound to look at brain blood flow. We can do this during exercise testing. We're also using different vascular reactivity techniques in an MRI. So in this case, Ethan and Aiden are using carbon dioxide delivered through something called a Douglas bag. And a participant will breathe this in while they're in an MRI. And we can look at how the vasculature in the brain dilates and responds to the stress. It gives us an indication of blood vessel health in the brain. So beyond just looking at resting blood flow, we get a sense of vascular reactivity as well. And then recently, my group was invited to go to a high altitude expedition where we looked at the impact of ketones and exercise on brain blood flow. So we're not just applying these stressors in a standard physiological lab, but taking some of these trials as well to different extreme environments to see how the system responds to different stressors. I'm going to give you just a really brief overview of the exercise side of my interest and transition towards ketones. But the way I view exercise is as what I call a neurogenic trigger. So you perform a single bout of, say, 20 minutes of exercise, or like Alexi, you run up the stairs in a big sprint snack, and you have a number of these exercise stress responses that are beneficial for long-term adaptation. So increases in metabolism and brain blood flow, a change in these neurotrophin signaling, which help to retain adaptations, increases in lactate, which is another alternative fuel for the brain. And then what you get also is an improvement in cognitive function for up to 1 hour post-exercise. So exercise is this potentiator of brain function if you do it even once. Okay, there was more to here. I have some other stuff that's not going to show up. What happens when you have acute exercise trigger is it leads to cellular adaptations that start to promote brain plasticity. So genetic expression of plasticity genes get upregulated. And if you repeat these bouts over successive days, weeks, months of a training period, you get these long-term adaptations that we're familiar with. So increased brain volume, greater brain connectivity, greater mood, resilience, and cognitive function, and then protection against neurodegenerative diseases long-term. And in my work within this field, I've done a lot of work looking at this neurotrophin called brain-derived neurotrophic factor. It's thought to be like miracle grow for the brain because it really directs the growth of new brain tissue. It facilitates learning and memory. And what you can see is when you do a single bout of exercise, this hormone goes up and it goes back down. But if we continue this repeated bout, we can start looking at how we improve brain health long-term. And we've also used this paradigm under different types of energetic stresses. So not just exercise. So we found that fasting also upregulates BDNF. You can use a single forearm hand grip exercise because it's a sympathetic stimulus. It's going to evoke some of these stressors, increase BDNF as well. And then also things like whole body heating. So there are different ways to get the body into this kind of neurogenic state, we believe, that go up beyond just traditional exercise that we can use for the purposes of brain health improvements. So for today, I'm not going to focus on this, but I want to give the context through which I think about this work. First, I'm going to talk about aging as a nonlinear phenomenon, and then introduce the cerebral energy crisis that's been established by Stephen and his team. I'm going to show you some work that we've done in the way of exogenous ketone supplements, and I'll give you some final thoughts of my ideas for a vision in the ketotherapeutic chair. So take-home points first. Exercise is a cognitive stressor. It's not just a physical stressor, and it stimulates all these improvements in brain plasticity. It does this also by increasing alternative fuel sources like lactate and helping with brain metabolism. And I think if we really want to incorporate exercise as an intervention for brain health, we have to think multimodal. So I think exercise is going to be the stimulus to prime the brain. And now you need to use other behavioral interventions that are going to help with that brain plasticity as well. The second take-home point is that as we get older, our brain glucose metabolism and brain blood flow start to decline, and these are dementia risk factors. We know that ketogenic interventions might be able to support the brain through metabolism and through brain blood flow. And so far to date, there's mixed evidence about how these ketone supplements work with brain health. I think the evidence from Stephen's group is quite compelling, but with the emergence of new supplements, we need to be doing a bit more work in this space. So the title of my talk was Building Better Brains. And I think if we want to think from an aging perspective of what ingredients go into a better brain, we go beyond just physical activity. We need to think diet, cognitive stimulation, social engagement, sleep is a huge one, and then reducing cardiometabolic risk. And if we had a recipe, I think it would look like this. So number one is reducing those cardiometabolic risk factors. We know that hypertension, obesity, and diabetes account for a large portion of modifiable risk factors for brain health and dementia. We also need to build neural reserve. So like skeletal muscle, we want the brain to be bigger and stronger to withstand stressors as we get older and to have a greater trajectory for aging. And then finally, we need to fuel the brain to do this kind of work. So I think when we think about aging and changes in our physiological and biological systems, the dominant model is that as we progress with chronological age, we show this gradient linear increase in our biological aging as well. But there's really strong emergent evidence of this more life stages approach to aging. And that is, as our chronological age advances, we have these epochs or these moments in our aging trajectory where we have big increases in our biological age. And so this could look like something like puberty, which is a big epoch, menopause, social stresses. But I also think in this range, where we have increased cardiometabolic risk factors, we could have a turning point where something like the onset of diabetes or hypertension leads to an acceleration of aging. And my real point here is that focusing on what is happening during these epochs is very important for early intervention to have people on a more optimal track for aging. So the way that lifestyle contributes to brain health, insults or helps, the way that this contributes to maybe neurodegeneration disease is things like diabetes, obesity, and hypertension, impaired brain blood flow metabolism. It causes brain insulin resistance. It can lead to altered immune function, and it also reduces the expression of these neurotrophins. So we obviously can't pick our parents or our genetic material that we're born with, but we can address these lifestyle contributions and focus on these factors specifically that are going to impact brain health. So I want to show you some data that really supports this idea of the life stages versus the gradual increase in brain function. So this is a recent trial that was published. It used data sets from the UK Biobank and a couple other big databases. And the researchers asked, what does brain aging look like over this midlife to older age lifespan? Just to orientate you, this brain network instability metric is an fMRI-based measure of what they call brain age. A higher number means that the brain is less connected and less complex. So this indicates more aging. What the authors found was that brain aging has these transitions, especially at the ages of 44 years old and 66, where they see an acceleration or worsening of this metric. And they looked to see at these epochs what explains this acceleration. And they found that people who had higher glucose and higher blood pressure at both of these points tended to have more acceleration in this brain network instability. So key points of intervention. They then did a really neat analysis as a follow-up. So they took this brain map of these regions that showed more of this instability, so more brain aging, and they overlaid it with a map of the human brain gene expression. So they looked at regions of the brain that had different genetic expression to see if genetic expression was linked to some of this dysfunctional brain aging. And just to orientate you to this, they arranged it through genes that regulate energy metabolism, vascular function, inflammation, and just the control. And what they found was it was the genes that regulate metabolism that were most aligned with these dysfunctional areas of the brain. So if we focus just on the top figure here, what this graph is saying is that people who have higher expression, brain regions that have higher expression of the GLUT4 glucose transporter tended to have worse indices of brain function as they got older. So that areas of the brain that are more reliant on glucose through an insulin-mediated mechanism tended to have more of this dysfunction. Conversely, if we look at this panel here with MCT2, which is a lactate and glucose or a lactate and ketone transporter, regions of the brain that had higher expression of MCT2 tended to have better indices of this brain network measurement. So the implication here is that there is clearly a vulnerability towards glucose-specific pathways in the brain and also maybe a protective effect of the ketone-specific pathways. Now this is cross-sectional. It is not looking at any sort of intervention, but it is still providing some evidence for this phenomenon. And this brings me now to the work done here at Sherbrooke and by Stephen's group that is real establishing this glucose hypometabolism as we get older. And so you might have seen this figure many times, but essentially we see that over the gradient of normal cognition, mild cognitive impairment, and Alzheimer's disease, there is this progressive and profound glucose hypometabolism that exists in the brain. So certain regions of the brain cannot use glucose as effectively, and it probably underlies a lot of the cognitive issues we see in these conditions. Is that what I sound like? But if we look at acetoacetate, so ketone metabolism across the spectrum, you see that ketone metabolism is retained in the brains of people with MCI and Alzheimer's disease, and that we actually see compensation in regions of the brain that have this glucose hypometabolism. And I think one of the most exciting and interesting findings from this work that's very reproducible is that when you have more ketones in the blood, the brain will take that up in a linear proportional manner. So there's a real potential for application by adding more ketones into someone's blood, their diet, whether exogenously or through diet manipulation, that can help raise ketone body levels. And this is where the brain energy gap really comes in. And again, something that's been pioneered by Stephen and his group. So we know with aging, we have this reduction in brain metabolism as a normal part of aging. But then if we look across the transition towards neurodegeneration and aging, this gap becomes widened where someone's brain metabolism is significantly lower. And the proposal here is that given that ketones are still taken up and metabolized by the brain, despite glucose hypometabolism, we'll be able to fill this gap through ketogenic interventions. And so this is where the advent and the application of oral exogenous ketones come into play. So there are a number of emerging different products on the market right now. The stuff used by Stephen's group was medium chain triglycerides, so MCT oil. There's also ketone salts, which can be used in infusion studies, as well as an oral product in a sachet that's reconstituted with water. And then there's the ketone esters, which I've been using primarily. Now, I can't speak to all of these techniques because I've really only focused on the ketone ester. But one of the attractive features of this product, if you notice the BHB levels that you can achieve with these products, so the ketone ester actually induces rapid and profound nutritional, or I guess exogenous ketosis. There might be some drawbacks with this, but this is a product that we have used in the past. So if we're looking back at the recipe to build better brains, we can fuel the brain by adding more ketones into the blood. There is evidence as well that beta-hydroxybutyrate is a signaling molecule that actually can upregulate some of these pro-plasticity pathways. And finally, there's application to help reduce cardiometabolic risk factors using ketone supplements themselves or by having dietary change. So what I'm going to show you now is some data from my postdoc work with Dr. John Little, where we tried to directly address the cardiometabolic risk factors, and we found there might be some benefit for the brain as well. So in this crossover trial, we had 14 adults with obesity. They were in a double-blind randomized controlled manner. So they took a placebo or the ketone and switched conditions. They maintained a normal diet, and we actually provided them with all their food throughout the intervention. So it was very controlled nutritionally. And they supplemented with this ketone monoester by HVMN 3 times a day. It was a relatively small dose, maybe about 20 mils of the supplement per dose. And this is just an indication of who was in our study. So average age was about 56, and the average BMI was almost 34. They had normal glucose, and they had elevated blood pressure as well. But the presence of obesity was the main criteria to be in this study. And we did a number of different assessments in this trial. So cardiometabolic health, brain blood flow, cognition, and some immune function as well. I'm just going to show you some of the cardiometabolic and the brain health as well. The real purpose of the study was to try to give people ketones before they had a meal, because there's been data to show that if you take ketones prior to having a meal, it actually lowers your blood glucose after the meal. So we think this might be a way of protecting cardiovascular health. So one of the first findings was that we actually do see a reduction in blood glucose after a meal, as well as over 24 hours when someone is on this ketone diet for the 14 days. We also wanted to measure the peripheral vascular function, because we know that these spikes in glucose after a meal can lead to long-term impairments in someone's cardiovascular health. We use a technique called flow-mediated dilation, or FMD, where we essentially inflate a cuff around the forearm for 5 minutes. You do an ultrasound of the brachial artery, and then you release the cuff to watch how the vessel dilates in response to that shear stress as an indicator of someone's vascular health. This is meant to be possibly a surrogate for someone's coronary artery health as well, so it does have some clinical utility. And if you can see here on the graph, we have the pre and post placebo and pre and post ketone. Following ketone supplementation, there is a significant increase in this vascular health measure in these people. So whether it's connected to the glucose lowering or something ketone specific, we're not too sure. But this is a very encouraging finding about the potential for ketones to improve cardiometabolic health. As it relates to brain health, which is my primary interest, we also looked at brain blood flow, looking at arteries in the neck, and we found significant increases in brain blood flow in 2 different vessels leading to the brain. Concurrent to this, we found improvements on a cognitive test that includes processing speed, working memory. It's a multi-domain cognitive test called the digit symbol substitution task. And what was really interesting to me is that when we looked at the relationship between someone's improvement on the cognitive test and their increase in brain blood flow, there was a significant positive relationship between these two. So this is not direct evidence, but it does suggest that maybe improvements in brain blood flow are connected to these improvements in cognitive function, and definitely something worth following up on that we're currently doing. Now, what's interesting about this is we did this in 2021. This has now been replicated in a trial looking at adults with obesity who undertook a ketogenic diet compared to a standard diet. In this trial, they found a significant increase in people's brain blood flow following this 3 weeks of a ketogenic diet compared to standard diet. And they found that the concentrations of ketone bodies in someone's blood was related to their improvement in brain blood flow. So again, there could be a role for brain blood flow in this situation. Finally, I mentioned that growth factor BDNF before. I'm just going to flash it up here, but they found significant improvements in this factor as well. So there could be this fueling the brain, helping vascular function, and improving the neurogenic situation as well. The reason this is important to me is we're seeing these long-term changes, but if you look at infusion of ketones in an acute manner, so you give someone an infusion over, say, 3 hours, you see profound changes in brain physiology. So in this group, this is older adults who had an infusion of ketones for 3 hours. They found a reduction in brain glucose metabolism, suggesting that ketones are offsetting that because there's no change in overall brain metabolism. And what's really compelling is they had up to a 30% increase in brain blood flow in that 3-hour period. So the brain is very responsive to an increase in these ketone bodies and increases perfusion and delivery of oxygen to the brain. So this left us wondering, we have infusion techniques, we can't do this to people in the real world, but we have these supplements. What happens when you supplement somebody with an exogenous ketone? And so my master's student, Aidan Rorick, wanted to examine this, looking at a dose response study where we took young adults and we did a crossover trial. They were given 3 different doses of either a placebo, a low dose ketone, or a high dose ketone. And they essentially just rested across a 2-hour period, and we measured what happens to brain blood flow and other parameters. What you can see here is that over this time span, we were effectively able to create 3 distinct conditions where ketone bodies were the highest in the high group, about half the amount in the low group, and there was no change in the placebo over this time. The main outcomes I'm going to show you today is what happens to BDNF as well as brain blood flow. So the first thing we found was that the high dose condition increases this neurotrophin BDNF, whereas we see no changes in the low or the placebo. And then if we look at the relationship between the cumulative beta hydroxybutyrate concentrations in the blood compared to the change in BDNF, there's a significant positive relationship between the two. So there's something about the beta hydroxybutyrate or the ketone ester that is related to this BDNF response. And I can speak more to that shortly. The main outcome that we're interested in is how ketones affect brain blood flow. And contrary to what we saw in the infusion trial, we saw that brain blood flow actually was significantly reduced in a dose-dependent manner when you give somebody ketones. So you can see that at 45 minutes, both the low and high dose condition are about the same. And then we see a significant reduction even further in the high condition after 2 hours. So not what we expected. And then if we looked at the relationship between these two, we can see that this is basically peak beta hydroxybutyrate was related to someone's reduction in brain blood flow over time. And you can see a real gradient between the high and the low conditions. So more ketones leading to more reductions in brain blood flow. Not great. Well, not what we expected. We measured cognition, and there's no change in cognition. And this is a pretty robust finding that if you lower brain blood flow within a physiological range, your brain does quite all right on cognitive tests in that acute period. I'm sure over a long-term period of time, it is not good, but that is to be discussed. So what's going on here is that ketone monoesters provide a weak acid. Beta hydroxybutyrate is a weak acid. It increases hydrogen ion in the blood, which then gets buffered by bicarbonate. And through the bicarbonate buffering system in the blood, we have an increase in carbon dioxide production. This leads to more ventilation. You start breathing more. And in turn, you actually reduce carbon dioxide in the blood. So why is this important for brain blood flow? Well, brain blood flow is highly sensitive to carbon dioxide in the blood, such that a small drop in arterial PO2 leads to a pretty sensitive drop in brain blood flow. And sure enough, if we look at the relationship between beta hydroxybutyrate and someone's CO2 change, it follows that same brain blood flow profile. So this is probably due to the acid stress of the ketone monoester product and not the ketones themselves. We then followed this up in a study by master's student Claudia Young. And a really simple study, she had people take a ketone monoester, and then they had a face mask on. They either breathed room air for an hour, which allowed their, it's going to work. It allowed their CO2 to drop, as we've seen before, or they would deliver carbon dioxide in a consistent manner over this hour period, and we maintain their CO2. So essentially, if we keep CO2 from dropping, what happens to brain blood flow? And I think what you expect happened. In the free breathing condition, we basically reproduce our findings that brain blood flow was reduced with ketone ingestion. But when you maintain carbon dioxide, you're able to actually prevent this drop from happening. So this could be a potential strategy where we need to increase buffering capacity of the blood if we want to mitigate that drop. Also, that acid stress might be beneficial long-term because we know exercise causes acid stress, and maybe this leads to cellular adaptation. So I'm just noticing my time right now, and I have some slides I was going to show. I'm going to skip past them, but happy to chat about it. Essentially, we did exercise and ketones, but for the purpose of this talk, I'm just going to skip past it, but I'll give you a brief summary. Ketones are lowering brain blood flow because of this acid stress, but cognition's not being improved. Again, it could be beneficial, we're not sure. There's an increase in BDNF, and I think this is due to the same stressor. We're probably having chemoreflex activation leading to a bit of a stress response, similar to exercise. And if we prevent these reductions, it is leading to an improvement in brain blood flow. So what I think the next piece of work we have to do now is to see, does this acid stress matter for adaptation, or is this just a side effect of the product that we're using? Okay, so this is the acute young adult work that I've been focusing on. I'm going to now show you our clinical arm and how we're applying this in older adults. And specifically, we're trying to identify red flags for intervention. So we know that when people progress towards MCI or Alzheimer's disease, they probably have been enduring decades of drops in metabolism and brain blood flow and maybe brain atrophy. What we're focused on right now is this region called subjective cognitive decline, where people start to have the onset of cognitive symptoms, but if they went to their doctor, they would not be diagnosed with any sort of cognitive issue. We think that there is still reductions in brain blood flow and metabolism happening during this stage. There's clear measurable dysfunction with fMRI, and there are clear sex differences at this point. So we believe this might be the first behavioral manifestation of dementia risk and probably a really important time to intervene on somebody. The next red flag is what we call dual task functioning. So if you imagine someone is walking, it takes some energy to do that, and now they're walking and performing a cognitive task at the same time, and that requires even more energy. Now, if you have somebody who has hypometabolism, we have reduced capacity in general for energy production, and we're operating at a lower status. So this kind of a task is going to stress the system even further and maybe show impairment. So we now are trying to use this SCD model as well as dual task function to really probe at someone's brain function to see if we can actually see differences before and after ketone interventions. So in a pilot trial run by my former postdoc, Dr. Jeff Coombs, we asked the question, how do ketones impact brain function if we use fMRI to visualize this? These are people who had subjective cognitive decline. And it's a very simple protocol. They did baseline measures of cognition. They took a drink of a ketone or placebo, and then we measured brain function using fMRI. We looked at functional connectivity in the default mode network. We looked at some resting state measures of brain activity, brain blood flow, and cognitive function. And just to give you a sense of what the ketone levels in the blood look like, this is the 30-minute mark, which is when somebody entered the MRI. And then the 90-minute mark is when they exited. So you can see that ketone levels were quite stable throughout the entire imaging session. So the first thing that we saw was that after only, I guess it was 90 minutes post-ketone ingestion, there are significant improvements in cognitive function on that digit symbol substitution task, which I showed in a previous trial as well. We did not see changes in Stroop task performance or in the task, MBAC task of working memory. So there might be some sensitivity to the DSST specifically that we're not seeing elsewhere. Very interesting though, is that we were able to reproduce the same finding that brain blood flow is being reduced. Just as a note, this is half of the dose that we gave in the high dose of our previous study, and we're still seeing reductions in brain blood flow. Despite these changes in brain blood flow though, we looked at a metric called the fractional amplitude of low frequency fluctuations, or FALF. This essentially is an indice of spontaneous brain function in the low frequency domain, and it is thought to maybe be associated with changes in brain metabolism. So it might be positively related to that. And what we can see is that after the ketone ingestion, areas of red indicate more of this FALF or spontaneous brain activity, and blue indicates less. So there might be an increase in some of these surrogates for brain metabolism, whereas other regions show reductions. We also looked at functional connectivity in the DMN. So this is the strength of connection between spatially dispersed networks that are integrated doing the same task together. Specifically, the DMN is really important for memory processing, self-referential thinking, introspection, and overall brain function. And this might be one of the first regions that show hypometabolism and impairment in Alzheimer's. And so we think it's a relevant metric to pay attention to in SCD. What we found was there's significant improvements in this functional connectivity in different regions of the DMN. So the precuneus, medial prefrontal cortex, and parietal lobe. So the takeaway for me here is we're seeing improvements in cognition after a single dose, despite reductions in brain blood flow. And we're seeing some metrics of greater connectivity and maybe brain activity. So there could be a change in efficiency, possibly in terms of how the brain's working with ketones. It could be a restoration of metabolism back towards maybe a more normal state. But again, this is a single dose, so it's hard for us to really make inferences about this. So we have taken the same protocol. We've looked at SCD in this last study. Our next question now is what happens to people with mild cognitive impairment? Because we know this transition, especially in SCD, is quite heterogeneous, and it can take a long time for people to transition. When someone has MCI, there's overt clinical diagnosis. So I partnered up with a group in Basel, Switzerland, and we're running some pilot trials. We're basically harmonizing our data collection in Hamilton and in Basel and we want to run a multi-center RCT where we have people with MCI or SCD in a long-term supplementation trial. So this is our first steps of showing that we can work together and harmonize our protocols. So we've collected to date 11 out of 18 people. And what we found was that there's a significant increase in this one metric called REHO, or regional homogeneity. This is meant to be an indicator of local, very focal synchrony between regions or within a certain region of the brain. And work from here at Sherbrooke, I think this was Kevin Whittingstall's group, and Stephen, you're involved in this as well, showing that the cerebral glucose metabolism is positively related to someone's REHO score. Now, this is a bit challenging because we're giving people ketones and we're trying to make inferences of what's happening to glucose metabolism, but I would interpret this to mean that maybe there's some changes in brain function as well. So stay tuned for this. We're hoping to collect the final participants this month and get a better sense of what's going on. Okay, and just to wrap up, we have an ongoing trial right now where we're actually taking these acute paradigms and looking long-term, or I guess over 14 days, of what's happening in people who take ketones. So people are taking 15 grams of this ketone ester 3 times a day in a placebo-controlled crossover design. They all have SCD between the ages of 60 and 75 years old. And our final numbers we're recruiting for is about 46. We're measuring a number of different outcomes, including heart rate and blood pressure. We're doing that dual task functioning, cognitive tests, and then a variety of MRI measures as well. And I've already shown this picture, but this is one of the vascular images that we're showing. And just to give you a sense of what's the adherence in a protocol looks like already, this was done by a thesis student named Zena. We can see that over the 9 participants and 5 that are still ongoing, adherence to the protocol has been very high so far. So most people in the 100 to 90% range over the 2 trials. Okay, so just to summarize what's been going on with this work and what I think needs to happen next is we know that reductions in brain blood flow and metabolism are risk factors. We can use ketones to support brain metabolism, and I think that we're still trying to understand what these findings mean. We need to have a more long-term trial to start looking at these behavioral outcomes in these folks. Also understanding sex differences and individual response phenotypes. And then a big thing for me is combining ketones with other interventions to look at real-world applicability of lifestyle intervention. And then a big part too is this is a very short period of time. People are adherent to the protocols, but it's only 14 days. How do you ensure long-term behavior change for sustainable improvements that people will have autonomy and want to do over a long-term period of time? So ketones on their own are having all these positive impacts in the brain. We know that exercise has these impacts as well. We could take exercise, we could add social engagement, cognitive training, stress reduction. These are all going to intersect with these important systems that are impacting brain health. So again, I think the next step is going to be putting these all together in a manner that allows us to look at long-term change in the brain. Okay, so to wrap up in the last couple of minutes here, this position of keto therapeutic chair has really been pioneered, spearheaded, and created by Stephen, and there's an excellent platform here at UDS. I think where I see myself fitting into this is bringing in this mechanism to clinical approach and starting to answer more of these questions in a way that allows us to apply it in real time. I think identifying some of these physiological signatures of who responds and why are quite important. Also understanding doses as well. And I think extending keto therapeutics beyond just a single ketone trials, I've said before, to other types of interventions. I think sleep physiology, cognitive training, social gerontology as well. We know that people have isolation and issues of loneliness and aging. We can't be thinking of just addressing brain health in such a medicalized fashion. I think developing long-term RCTs is going to be a big thing. So to position UDS and CDRV as a global leader in intervention strategies using ketone approaches. I have really strong interest in using sleep interventions, which I'm happy to speak to later, because we typically think of these waking behaviors in isolation. It's a very catabolic state where we're using energy or breaking down energy. But during sleep, we have the replenishment of energy systems and the removal of waste. So we need to think as well about the other side of the coin. I'd also like to build a data repository where we actually get to take these small RCTs, pull it together in a big harmonized manner, and start to explore what's happening in bigger data sets. And then I think also developing an international hub for trainees who come to Sherbrooke looking to learn keto therapeutics and lifestyle interventions with PET imaging and other resources here as a way to draw trainees as well as collaborators into the network, and also to build these big RCT networks. And then finally, I have a couple asterisks here because I don't speak French very well and I need to really learn that. But community engagement, health equity, and user-designed thinking is critical to all of this. Again, we can't just medicalize these interventions. We have to think about the end user. So having participants be the designers, co-designers with you, thinking about access to people who have either geographical access limitations or socioeconomic limitations. And then, of course, knowledge translation and community partnerships are critical. So I know a lot of this work is ongoing and happening here right now. And I think that there'd be a real synergy in that regard as well. So thank you for your attention. I'm happy to take questions.
Super, Jeremy. Merci beaucoup. Alors, c'est votre tour. Helen.
Thanks, Jeremy, for this mic. Oh, thanks, the microphone. Thanks for your nice talk. I have many questions, of course, but I'll limit myself. My first question is, you've been recruiting male and females in all your studies, but you haven't spoke about it here. So did you see any differences in any blood flow or BDNF or the other?
So yeah, it's a great question. Yeah, I did not mention specific sex differences, but this work is ongoing. So I showed you the trial where we did a dose response study and then the follow-up of the CO2. We are now combining those data sets because we use the exact same measures and same time course. And a summer student is starting to look at sex differences in that work. And then in terms of our older adults, that's analysis that we plan on running as well.
But you haven't started.
We haven't done anything, and I can't.
Any hypothesis?
Speak to that too much. It's a great question. I feel like women might be more responsive to the ketone intervention in aging. I wonder if maybe like the onset of menopause is impacting brain metabolism in a manner that is more profound. Where men, you have that gradual decline, but with the onset of menopause, you might have that life stages kind of acceleration.
So yeah, I was very preoccupied by your curves because Melanie and I were looking at it, trying to look at where we're right now.
I know,
yeah, 44. Yeah.
And I know with Melanie, like we've been talking about this too. You have that perimenopause, postmenopause transition. Yeah.
You want to talk about that?
And we know it's. But yeah, the vascular side as well as the metabolic side, I think in the women are probably more sensitive to that change. So I'm thinking if we're going to see a positive effect or an interaction with the ketones, I imagine women would be more sensitive to that. The age ranges here are pretty well matched, so we're not seeing big age discrepancies in our sample. So yeah.
Thank you.
Catherine, est-ce que tu as des questions en ligne?
Non, j'en ai pas en ligne, mais il y en a une.
Non, OK.
Il y en a une en ligne pour la prochaine.
Hi, thank you so much for a great presentation. Can you tell us a little bit what you saw in terms of neuroinflammation?
It's a great question. So we haven't measured neuroinflammation.
So
yeah, in the other trial, we looked at immune cell function from peripheral mononucleocytes, and we saw that immune function was actually improved. So the master student at the time, Helena Neudorf, isolated PBMCs and then treated it with LPS and looked at activity of caspase 1. And we saw a reduction in that signal. So it seems like in that case with the adults with obesity, there was improvement in immune cell function. But in our trial, we were not seeing any changes in like inflammatory cytokines. and haven't looked at anything from a neuroinflammation perspective. I do think though that at least from the redox perspective, you think of oxidative stress, so not immune cell function, but ketones do tend to shift that towards a more like antioxidant profile. I think you see reductions in oxidative stress. So I think from that perspective, you might see some protection. From the neuroinflammation, I'm not sure.
Okay, and my next question is, do you have like a feeling of what genetically might predispose somebody to be a responder versus a non-responder of this approach? Because I've seen this approach been, investigated in people with dementia, people with schizophrenia, people with autism, and they all have like similar genetic markers also. So
Interesting.
I'm kind of interested in the
Yeah.
possible underlying.
What do you have? Like, are there specific markers that you can share? Or
Well, they're just all over the
Okay.
map, but there's like, several of them
So-
that
so
are common.
where my mind goes is there's so the MCT monocarboxylated transporter is important for moving ketones and lactate and other substrates into and out of the brain. And there are polymorphisms of the function of that transporter. There's been a trial recently that looked at intermittent fasting and people's responses in the brain to that. And they found that people who had this polymorphism, which negatively impacts function, tended to have like no response to the ketone intervention or the fasting intervention, whereas those who had the other version of the gene that was like the proper function were the ones that showed improvements in different brain health measures. So we're going to be assaying for this and looking at some genetic factors as well. So I'm really curious about that one. The other piece that I didn't explain is we have a collaborator who does proteomic analysis. And so we'll be doing blood proteome of organ-specific signatures. So from that, it's more of exploratory. I don't have a hypothesis. We might get some signatures of the proteins being expressed that might then give us some indications of possible genetic influences too.
Thank you.
Thank you, Jeremy, for the nice talk, as always. So quick question. So I think one of the main, component of your research is beta-hydroxybutyrate being, this important molecule. And there's multiple ways of increasing BHB in circulation. You talk about exogenous ketosis, but, there's intermittent fasting, ketogenic diet. So if you had to choose, or can you talk about, you're using one model, but you think one model would be better over other? Would you combine them? And the pharmacokinetics of all of them is different. So
Yeah.
from a brain perspective, what would be your best option? Or just
Yeah, it's a great question. And I guess the focus is neurodegeneration. And yeah, I feel like if we think about sustainability and something people can follow, I think reduced carbohydrate is probably the move. So you're really going to address a lot of the cardiometabolic risk factors with that, as well as having just less insulin resistance in the brain. So I'd be focusing more on the metabolic function of the brain from the peripheral inputs. I could see a situation where with reduced carb, you have some sort of a supplementation to complement that piece. So you still can increase blood ketones without having to do severe restriction or potential dietary manipulation that with poor adherence doesn't work the same. And this is where my knowledge and experience is kind of at its limit, because I haven't used MCTs or ketone salts, and then that'd be a discussion with the experts. But I do think that low carb is probably the move, as opposed to any more severe restriction.
Yeah.
What are your thoughts?
No, so I think you're right. I think combining different approach might be the way to go, and like long-term reducing carb, as Stephen done previously, instead of doing a full keto with less than 50 grams a day might be better for older adults. I think, as you mentioned, probably combining with exogenous ketone, whether it's MCTs or salt or ester. This is to be determined, but yeah.
And I think the exercise piece matters here too, because I didn't mention it, but we know lactate is very similar to ketones in terms of when arterial lactate increases, the brain takes it up in a linear fashion. And there's good evidence from humans that higher lactate during exercise is related to better cognitive improvement in the post-exercise period. So we have different ways in which we can influence brain metabolism. And I mean, the spin-off of exercise is all of the cardiometabolic benefits. So I think combining those would be probably essential.
D'autres questions? Marie?
In your SCD study,
Yeah.
were you able to correlate the BDNF levels to the changes in cognitive scores?
So we didn't take BDNF blood in the acute SCD study. We will be doing that in our 14-day RCT. So we haven't looked at that yet. I mean, so the BDNF story is a bit confounded in my opinion, because blood concentrations do not necessarily mean brain concentrations. It's used as an indicator of, I think, this stressor pulse, where like BDNF goes up because of a stressor, and it also goes up in the brain. It maybe goes across the blood-brain barrier, but I think it's tough to look at blood levels and try to make inferences about acute cognition. It might be something better for like a longer-term intervention where you see like an adaptation kind of correlating because of, yeah, both systems are improving together.
Speaking of subjective cognitive decline, it's a heterogeneous condition.
Yeah.
My understanding is it's essentially self-declared,
Yes.
with the paradox being that if I don't think I've got a memory problem, then I don't have a memory problem. People around me might not agree.
Yeah.
How did you confront that
Yeah.
challenge?
That's a tough one. And that's one that is, so the Alzheimer's Association recognizes SCD as what they call stage 2 of the dementia continuum, because there is something observable if somebody self-declares it. The other piece is a spouse, a partner, or someone who's close to a family member might notice it and also self-declare it on that person's behalf.
Okay.
So those criteria are both reasonable, but because it's so heterogeneous, you could have the worried well, where someone is noticing that they're getting older and their memory is changing, and but it's not because of an underlying issue. It's a natural age progression, whereas those who have actually a rapid decline. So I think the challenge here is you need actually probably bigger numbers of a sample size to actually truly capture a signal within a heterogeneous group. But based on the guidance of the Alzheimer's Association, I think it's still worth probing. So to answer your question, the questionnaire is like a single question, really. Like the retrospective prospective questionnaire is like, have you noticed a change over the last 12 months? There's other ones that say over the last 5 years. It's very limited. And then there's something called SCD plus, which is more SCD. And that's if you have these changes that are specific to memory. So it's cognition changes is the original one, but if it's a memory change, then you're at greater risk. So all that to say is I think it's something that needs to be teased out a bit more, and probably finding if there are phenotypes within that group. And that's why we're looking at the dual task function, to see if we can start doing some behavioral tests within an SCD group to look at some of these manifestations of maybe advanced changes in the brain.
D'autres commentaires? Oui. Ça va coûter la prochaine fois.
Have you noticed some side effects? You know, like we hear like kidney stones and things like that. People need to be monitored.
Yeah, we haven't seen that. We are doing like blood monitoring, and there have been no changes in any of the panels. We had one participant who had to stop after a day because he had like bad acid reflux. And I haven't seen that before. We do like gastrointestinal distress questionnaires in all of our studies, and you might occasionally get some belching or a bit of bloating, but it's typically not that bad, especially at the lower doses that we're using. But this person was quite sensitive, so they had to withdraw. But nothing is showing up in the blood panels at all.
On a encore quelques minutes, si ça vous tente.
In your data where you have these brains with the red and blue,
Yeah.
I know I'm not specialist at all, but what I've noticed is that there's an asymmetry. In some parts of the brain, in one hemispheres, you have red,
Yeah.
and in the other, you have blue. Do you think it's related to the task that was conducted, or it's really something that's going on in the brain?
It's a great question. So I think to clarify, this is called resting state analysis. So someone just lays there still and their brain is imaged at rest. So there's nothing task happening in real time. People have looked at these ketone products as being possibly therapeutic, but also maybe just being a metabolic perturbation. So you are introducing a new substrate into the blood, and now you're seeing how the brain cells and neurons are responding to this change in metabolic status. And so it could be a probe to see how metabolic function in the brain responds, but it might not be indication of like actual functional benefit per se. Like we don't know if this is just a knock-on effect of changing the physiology, or is this a true functional improvement in brain health? I think what we can look at is to see how the brain responds, whether it be uniform or asymmetric, but then run the RCTs to see is there an actual behavioral indication, and do these metrics improve when someone is in the fasted state, so no supplements, over that period of time. So it's a roundabout way of saying, I don't know, but I wouldn't read into too much if that's functional or if it's just noise at this point.
And how do you measure adherence? You were speaking in the last slides of adherence. So what's your measures of adherence?
We have people fill out a diary. We have them mark down their food and the time they take their supplement. And then at the end of the trial, we have them, we give them their ketones in these opaque bottles that are labeled A or B. And then they have to return all of their bottles to us. And so we get to see how many have been consumed versus not consumed.
And you think it's accurate? You can just drop it in the toilet.
It's true. I wonder, yeah. It's absolutely true. I tend to believe it because I don't think there's a pressure campaign of like, if you don't do this, I like to think people are being honest, but it's possible.
You don't measure any ketones in the blood.
No, they come in when they're fasted. So we measure them pre and post in the fasted state. And we want to keep it blinded because this is a blind study. So if we did some sort of a finger poke, they would be unblinded to their condition possibly. So there has to be a bit of trust in that regard. I do think though, for these longer term trials that are not 14 days, there might have to be some adherence check-ins and like making sure people are staying compliant too. Yeah. Yeah, it's like a. No, you're right, completely. Yeah. Yeah.
You need label. Yes. Random. Yeah.
Yeah, or just do an open label trial and people know what they take.
So quickly, it's actually great work and great talk. The only part I didn't quite like was that 44-year-old thing. That was not.
I'm 4 years away, so I'm like seeing that in a different perspective now.
I'm just wondering, at the end of your talk, you mentioned briefly other applications, including neuro rehab and things like that.
Yeah.
I'm just curious to have a few words on this from your point.
Sure. So I think of exercise as this trigger, right? We're going to have this pro-growth environment. We're going to have the brain maybe more turned on, ready, aroused, whatever words you want to use, that will be ready and receptive to brain plasticity. There's evidence from animals and preclinical models that combining exercise with enrichment or cognitive stimulation actually leads to greater neurogenesis in the brain. There's this idea that exercise will prime the brain for new neuron growth, but survival is contingent on reinforcing that with enrichment. The human models in terms of like exercise and cognitive training in older adults haven't really shown that quite yet. But I think in the situation where we can provide the brain with this new fuel source that it might prefer and that has signaling properties with exercise, we'll create this neurogenic or pro-growth environment compared to doing it just on its own. We've tried to pilot this with some TMS work with a colleague with the ketone ester. It hasn't gone too far because we're doing it in young adults, but I do think there's something to be said about priming the brain before you do some sort of rehabilitation through TMS, through stroke rehab or cognitive training. Thanks.
Maybe I could make the final comment, Jeremy. Thank you again for your talk. You talked about it as just now as a new fuel source.
Right.
And I just want to make the comment that
additional.
the brain gets used to ketones in utero. And it's during infancy that ketones play a key developmental
Mhm.
role. So it's not a new,
You're right.
I understand the context for saying it as new, but in fact, it's something we're trying to drag the older brain back into
Right.
health via the mechanism that made it healthy to start
And
with.
I'll say my last visit here, I was using that terminology very liberally, and Stephen gently provided this, and I completely agree. I mean, lactate is a good example. It is going to use these fuels flexibly when they're available to it. So would alternate be more appropriate? Additional?
Partner, complementary.
Complementary, yeah.
Merci beaucoup de votre participation.
All right, thank you.
Bon après-midi.